So, welcome everyone. We're super excited. If you're just joining, sorry for those who've heard me say this a couple of times. Feel free to put in the chat your location if you want, not your address, maybe your town, or just your state. If you want to put your duper's name and age, that's fine. First name only, please. Um, Certainly no pressure. And I actually have to pause 1 second. And we actually have two families that have joined right now. I see their names who have lost a duper. And so I want to also offer them a moment to even put, you're welcome to put your angel, you're welcome to put, because they're still part of your life. I think it's lovely that you've joined our call as well, to learn about where we're going. So feel free. I honor that and honor them. So thank you. But we are so glad to have you. We've had a great response to this conversation. Some people are like, I don't know what the heck we're talking about, but I want to hear what's happening with the science. Some of you may not know why you're here, but we will cover today some new initiatives that are really exciting. And Ryan has hit the ground running. If you haven't heard, she'll introduce herself in a second. And then we'll also give a moment to introduce the others on the call as well. So Ryan, I'm going to turn it over to you. One last homework, if any, or one more housekeeping. If anyone has questions, feel free to put it in the chat. We are going to tightly keep this on time. Most of you submitted questions ahead. But if there are things that pop up, if we don't get to them today, we're going to compile them. This is going to be a continued conversation. You may get the answer via an e-blast. We may do a follow-up, some type of call, but we will respond to questions. It just may not be today, but put them in the chat because if you have it, someone else may too. Ryan, I'll stop talking and I'll turn it over to you.
Hey everyone, thanks for coming today. I know it's tricky to make time in the middle of the day. So if you're eating your lunch, hopefully this is some nice background. But yes, I've been here for a few months now. My first official day was October 1st. Quick background, I am not a medical doctor. I have a PhD in biomedical science, and I have experience in genetics, epigenetics, neuroscience, physiology, all sorts of different things. In my past life, I was a professor and a grant writer. I ran a lab at Harvard Med School for a while. Not my lab, someone else's lab. So I'm very good at managing. But I just felt like I wanted to do something that made more of an impact. And I'm familiar with the Dup15q space because when I was in graduate school, my desk was very close to Dr. Stormy Chamberlain, and I always just loved her and was so impressed by her dedication to this work. And when James Fink, who's on the call now, approached me about joining as the Scientific and Clinical Initiatives Director, I really couldn't say no. It just all felt full circle to me. And I'm excited to be here now and then to take it a step further after meeting so many community members and researchers and physicians in the space. It just genuinely feels right. So I'm happy to be here and I'm really appreciative of everyone welcoming me so warmly. So, hey. I should also just pause and let Jackie and James quickly introduce themselves because I'm going to be kind of throwing it over to them from time to time because they have more experience in this space and also have more experience Jackie does as a parent. So just quick hellos.
I'll go real quick. So hi, I'm Jackie Vanderhoof. I'm a mom to Fiona, who is 3. She has isodentric form of Dup15q. And I'm a board member and the science committee co-chair.
Hi, everyone. Great to see you. I'm very happy to see so many people in attendance. My name is James Fink. I'm a scientist. I've been working on Dup15q since 2011 when I was a graduate student in Dr. Eric Levine's lab at University of Connecticut, where I worked not only with Dr. Levine, but also Stormy Chamberlain and met Ryan back then. So I've known Ryan for a long time, and I've known many of the people on this call for a long time. I serve on the science committee as a co-chair, and I'm also on the board.
Great, thank you. They also hired me, so that's cool.
You're big fans, right? You're big fans of them.
So anyway, I did send out a letter last month, which feels like yesterday, that kind of introduced myself a little bit and provided a very brief overview of the Learn, Treat, Cure initiative. in that text, I kind of highlighted that the main goal would be to expand the Alliance's scientific footprint to change the reality of a Dup15q syndrome diagnosis. So if you didn't get a chance to read the letter, totally fine. It lives on the website, and it's not going to be the last time you're going to hear from me. I plan to send out updates at the end of each month, starting in February, about any progress, any updates, anything exciting, anything that I think you would benefit from knowing. So sometimes it does look like a lot of words, which is why we also have been working on kind of making everything a little more digestible on social media. So Jackie, James, and Naka, they've been working together to kind of break down some of the goals or elements of each of the learn, treat, cure pillars, which I have here. So we're going to kind of shorten it to LTC when we talk about it or when we put it in writing, just to, shorten it. But essentially, our goals with this initiative are to learn as much as we can about Dup15q through innovative research, identify the most effective methods to treat the symptoms with limited side effects, and to develop disease-modifying therapy as a cure. So I want to pause here just to acknowledge that a lot of questions have come in, and we have been reading them regarding what treat means, what cure means. Learn is fairly self-explanatory. So with regard to treat, we are taking into account that Dup15q is not the same in every affected individual. We have 2 genetic subtypes, and we have a variety of symptoms that present themselves at various times throughout an individual's lifetime. So when we say treat, we're not limiting ourselves to just treating the seizures or just treating the GI or just focusing on autism-related treatments. We're looking at everything. We're trying to see the best way to treat the disorder for your child or your family member at a given time using what's available. And with regard to cure, which the majority of questions have come in about, in which we totally recognize is a very weighted term. When I first saw this program, I was like, cure, can you tell me what you mean by that? And then after talking to different members of the community, it became very clear that cure is not a one definition word. It means different things to different families. And so as an alliance, we have to recognize that, we have to account for that, and that's what we plan to do. So for one family, a cure might look like a 2-word sentence. For another family, a cure might look like several days without seizures. For another family, it might look like seizure-free. For another family, it might look like no debilitating GI challenges. So, of course, we would like to see a cure, meaning no symptoms at all, ever. And that is possible, but over time. I'm not going to tell you that it's going to be possible in the next year, but I cannot say either that it's never going to be possible. Science is advancing every day, and research is a collaborative space. Physicians are collaborative with each other, and the more information we have, the more potential we have to make a cure a reality for whoever you are and however it affects your family. And I think Carrie might have wanted to jump in here as well.
Yeah, one of the things I want to make sure the community understands is we took a lot of time to talk through this as an organization. Our board discussed it at length. We actually went to our medical advisory board, or our PAB, you may hear us call them. So it was really, we talked to some of our pharmaceutical partners, and we all kind of had the same, like, wait, cure, that's a strong word. We talked to other rare diseases. We talked to donors. We talked a we did a little bit of an analysis of all of our key leaders. And so this was not taken lightly. So if anyone's having that, like, this is a strong word, or you may come in this and go, this is a word that motivates me. And so we want this word to be motivational because, I think, Ryan, you gave perfect examples that it could mean something different to everyone. Even among the group of families that are on right now, all of you may put something, if we ask you, you'd put something different in the chat. If you were at the conference this year, we started talking about the word cure in some of our breakouts and saying, what does that mean to you? And so we want you all to know this was taken very serious, and something that our board is proud to be able to say, hey, we feel like this is the right word to use. So I just wanted to make sure that was clear to the community and how we came about this.
Yeah, we're very intentional. Okay, so what does this mean? So one of the things that we've been doing is trying to kind of categorize certain elements of the plan as a whole. So under Learn, there are a few tools and resources that we plan to use. So one of the things, which I'm going to talk about in a little more detail in a little while, is a natural history study. Natural history studies are very important for understanding the disorder, what it looks like, how it progresses, when the best time to intervene may be. So I am currently writing a grant proposal to submit to the FDA to get funding so that we can launch the first ever Dup15q specific natural history study. And I'll give you a little more behind the scenes in a minute. Mouse models. Everyone's been hearing about mouse models for a very long time. But sometimes it's just kind of presented as a throwaway term, like, we're going to make a mouse. Well, there's what? So there's a lot more to it than that. And I'm going to provide some insight on that again in a minute, but it's a very important tool in drug development and also in understanding disorders. Induced pluripotent stem cells, or iPSCs. That's James's wheelhouse. He's an iPSC guy. Do you want to talk a second about iPSCs, James?
Sure. So rather than using a mouse, which can walk around and move, but doesn't have human genetics, and there are differences, of course, between a mouse and the genes the mouse has and expresses versus a human. The induced pluripotent stem cells are a way to study human-associated genetics in a cell of interest. They're, of course, different body organs. Each of them is composed of different cell types, and it's not always easy to acquire a particular cell type, especially from brain, where you don't want to necessarily be doing brain biopsies on patients. So there's a technology that came out in the mid-2000s, I think 2006, has won the Nobel Prize for converting blood and skin and other samples into stem cells, which then can become neurons and other cell types. And then they're specific to the patient, and they have the disease and the genetics associated with humans and patients.
Thanks. And some other areas are the Ladder Medical Registry, which Carrie and Nika have been asking everyone to enroll in. That's a great way to learn about our current population, what is going on with our individuals at this time, so that we can make plans for what we should focus on, what we should try to target, and so on and so forth. And then, of course, through collaboration. For me, since I've been here, collaboration has been essential. By meeting with the community to learn more about you all and what you face and what's important to you, by collaborating with the researchers and clinicians who are either at the bench or in the clinic doing the actual work, and then also with other patient advocacy groups, where we work closely with the Angelman Syndrome Foundation, but I've also had the opportunity and the luxury to meet with other really impressive individuals running foundations in the rare space. And I've learned how they communicate, how they organize, how they mobilize. And they've just been an incredible resource for me, because they also have scientific and clinical initiative directors. And you can't learn anything by yourself. And so having collaboration and opportunities to meet and learn, it's been a plus. Treat, we have 2 clinical trials right now, and I'll talk more about them. Super excited about the Longboard data, but we will get to that. We also have our Dup15q clinics. We are constantly trying to improve, expand, refine, grow those. And in doing that, we are also able to bounce back and learn more. As we do this, we learn which medications are the most effective for which kids, which ones have less side effects, which ones have more side effects. And then we can circle back with our PAB, which includes physicians, to kind of learn about what they're seeing on the ground. And then, from word of mouth or from the website or from just medical releases, share that information with the community to improve the treatment opportunities and conditions for our families. Doing weird things with my hands. And then finally, in Treat, just an example, behavior interventions, because there was actually an analysis of some of the data from the Ladder registry, and a lot of individuals cited that there's a need for better behavior interventions. So we're learning so that we can treat, so that we can ultimately find that curative intervention. Initially, when I made these slides, I had jargon, just words like ASO, RNA. So that's not helpful. So what I did instead was I redefined it in a different way. I find that the best way to learn is to just keep redefining things in different ways because people have different learning styles. I should note I was a professor for a long time. So you just kind of keep reinventing the wheel until it rolls fast. So here for cure, you have to fix the mechanistic errors, meaning what's happening under the hood that is caused by the duplication. This can be going in and actually targeting the DNA sequence where the duplication exists, or looking at the molecular level, which is what is made by these genes that is too much, that is causing a problem. How is it causing a problem? How can we shut it down? And then the physiological level, meaning that within a cell or within a neuron, all of these gene products are being made, meaning proteins, RNAs, etc. They're interacting with each other, but they're not doing it right. So that's causing a problem. If we can get in there and break that down somehow, that could be a curative measure for this disorder. So how long is this going to take? Well, when I first joined, I was presented with a comprehensive list of goals, a wish list of sorts. So what I did was I sat down, I organized the information, and we kind of broke it down together into immediate goals, short-term goals, and long-term goals. Immediate is within a year, short-term, 1 to 3 years, long-term, 10-year question mark. So an example of some of the items on the wish list / initiatives that can be done immediately: launching this natural history study. I'm writing this proposal for the NIH. I'm also putting together a short form proposal for a few interested industry partners. Jackie, Carrie, and I have been on calls with industry partners who are interested in helping to support this natural history study because it's really important for them in their efforts to develop drugs for Dup15q. We want to increase the Roche trial enrollment, which those of you who have enrolled, thank you so much, because you're making a huge impact already. And if we continue to increase the trial enrollment, we can continue learning more and we can continue to move forward and then potentially identify other trials later as a result that we might qualify for. And then immediately increase funding for the scientific and clinical initiatives. Funding is important. In order to do all of these fantastic things, we need money to do them. So some of the ways that we're doing that is we're applying not only to these bigger federal FDA grants, but we're also looking at smaller foundation grants. We're tapping into previously untapped donor resources. Carrie is reaching out to all sorts of other individuals to try and help push this forward. And I'll give Carrie a second if she wants to talk a little bit more about that piece.
Yes, sorry, I'm getting texts and emails coming in and questions, and I'm like, oh, let me add this, let me add this. Yeah, you know, one of the things I think people often don't understand is the pace of what science progresses, like you're talking about, but then also how much the cost is that goes into this. I know we have people that go, how do we need all this money? We were a mom and pop organization, and one of the things that became very clear to me really quickly is how expensive science is. And so one of the things we want to make sure is, as we're talking about initiatives, as we put initiatives out, if there is something that you're like, I want to get engaged with this, I want to be part of the fundraising for this, I will customize anything for you. If you're like, hey, I want to run with this, I know people who may be interested in this science. We'll bring Ryan on a call, and James and Jackie and 20 of our board members, which we don't have. We have more like 12, but I'll bring on anyone to explain to a funder that you may be interested in us pursuing, because it is very expensive. And so anytime something strikes you as interested of an interest to you, and you want to know how to get engaged in the fundraising piece, feel free to reach out to me. I always tell people, if you can't find my email, you can send something to info@dup15q.org and reach any of us. So we need partnerships. It is definitely not something that we can do without the help of our community. And they have done amazing raising money. We wouldn't be where we are. NACA says this to me daily. There's no way we'd be here without the community fundraising like they do. So we got to continue that. So I'm happy to discuss opportunities. If your company wants to engage with us, if you have a company that matches your gifts, don't forget about that. If you're someone already donating, don't forget about utilizing that funding. If you want your gifts to go to research, just talk to us about that. So it's definitely going to take all of us. And I could do a whole webinar just on that if you can't feel my excitement. So thanks, Ryan.
Yeah, and Jackie has an important announcement that aligns with that as well.
Yeah, so I'm sure everyone has seen some of the social media posts about the gala. So the Dup15q Alliance is also having our inaugural gala March 2nd of, now we're in January of this year. And the funding is earmarked towards science and research initiatives. So it's really exciting that, you know, we're going to get a really, hopefully a good kickstart to a lot of these initiatives that Ryan is working on.
Yeah, I already signed up and got my plane ticket, so I will be there. So with this increased fundraising, we can then feed forward. We can develop what's called a seed grant program. So a seed grant is something that is applied for by a scientist or clinician, usually scientists, and they can apply for these seed grants, and they have to be specifically for advancing knowledge and development of treatments for Dup15q. With these seed grants, we can then tailor the research to our disorder, and then we can also bring in new researchers who can then grow. If you have a small pilot grant, you can do some preliminary experiments, answer a couple of smaller questions, and then use the data or the information that you acquire to apply for a big grant from federal funding, upwards of $500,000 a year for 5 years. So without the seed grants, you don't have the data, you can't apply for the big money, you can't answer the big questions. And I think someone who can kind of give a little more color to that would be James, because James is actually a recipient of a Dup15q seed grant way back in the day when he was a young person. Not that he's an old person now.
Man. So yeah, I think they're actually the really interesting thing, and maybe perhaps the most important thing about seed grants is that the outcomes of these seeds are unknown. I mean, they're called seed grants because the goal is to have them grow. And I can, as Ryan mentioned, speak specifically to a seed grant that the Alliance was able to provide me, and I was lucky enough to receive in 2015, so 8 years ago only. And then the course of these scientific advancements, as Ryan's describing, 8 years is a short time. That grant not only got me involved in Dup15q, and here I am many years later, still heavily involved and really thinking a lot about the science and trying to contribute as best I can as a member of the community. That was one way it's helped. The second way is it funded actual research at the time. Now that research has grown into two major things. The things we discovered in 2015 during the fellowship have led to differences we discovered in Dup15q neurons that are now being used by companies such as Quiver Bioscience and others to explore disease-modifying therapies. So that research now can be used for finding therapeutics. And then at the same time has helped Eric's lab develop further studies that build on that initial work, and has come out with several additional publications since then that have found other interesting things that may lead to therapies as well. So one grant, a seed grant given to a student at one period of time, has led to actually a really big growing and big impact in the field. And that's just one. And I hope to be able to give many others in the future.
Ryan, can I add in real quick as well? I think that, and I don't mean to be the fundraising pusher here, but sometimes I think people don't always know what's happening to the money we're raising. And this is a great example. And actually, there's a person on the call right now who came to us and said, "Hey, I want to do a scholarship on behalf of my child in my child's name." And they did that. They were able to help a lab. They were able to help a student. They've continued to do that. You may be on the call and say, "You know what? I want to do that. I want to raise X amount of dollars. This is what I can give." And so I say that because one of the best ways I've learned to advance research since I've been here is exactly what James is talking about. And now look at how this has trickled down, and now you've continued to spread the word. You've gotten the excitement built. You've stayed in touch with us. You've stayed excited about it. You've met the kids, and it changed your life, seeing the patients themselves. And so I want to encourage people to realize too that that's why it's important that we just keep this progression going. And that's why this community is so invaluable as well. So
Yeah. Some other short-term goals: develop better model systems, which iPSCs, mice, fruit flies as well are some great models. And then explore drug repurposing and trial enrollment opportunities. These things are ongoing. They're kind of always happening behind the scenes. As I mentioned, Jackie and I, we meet with different biotech companies from time to time just to see what they're doing, if there's anything that could potentially reach us, or if they're interested in modifying it a little bit, how we can kind of get involved in any way possible. And then long-term, obviously, a biological therapy trial, one of those curative disease modifying approaches. And at the end of the natural history study, another goal that we have is to better define the genetic subgroups between isodicentric and interstitial dupe so that we can understand the similarities and differences so that we can improve treatment for both groups, standard of care and treatment. So I look at it as what I'm doing now, what I'm doing next, and what I'm going to be doing for a while. That's how I have broken these things down, and I have a really intricate color-coded chart to keep myself on track, see where I am, what I'm doing, and where we are progress-wise, and who I need to lean on. The community has been fantastic, especially the board members, of coming to me and saying, how can I help? This is what I know how to do. This is who I know. Let me put you in touch. Let me help you do this. It's just been a really wonderful community so far. So all that's to say, how do we get started? Well, first thing we did was we established the content for each category because it can get a little bit murky. Conversations that we had were, well, is a clinical trial learning or treating? Where do we put that? How do we categorize that? And honestly, things might change. There might be a point where stuff that we've learned has moved into a treatment that eventually evolves into a real life curative measure. Identify initiative goals, what do we hope to get out of them, and construct a timeline for the rollout. James was really helpful with this in putting together a Gantt chart, which we were then able to present to the Dup15q board to tell them how we plan to do this, when we plan to do this, and with measurable outcomes for each of the varied stages. Again, within the board and the staff, we've identified roles and responsibilities to make this work, make it happen, and not be a conversation, but be an action plan. And here's what we're doing now. We're educating. We learn from each other. I believe in lifelong learning. Never stop learning, never stop asking questions. So by presenting this information here, this is not, I am literally a talking head right now, but in my real life, what I plan to do is continue to communicate with everybody. We're having the town hall. I sent out that first piece of literature. I'm going to continue to send the updates at the end of each month. We have the social media platform where we soft launched this. And again, this is all possible because of the community, because of the staff, because of the Alliance, because of the board. It is really a united effort so that we can make moves. We can make it happen. We've started to engage our industry partners already. We have a really nice tight-knit network with researchers and clinicians, and we're constantly expanding it, reaching out to people who published something one time that mentioned a duplication and saying, hey, you mentioned this one time. You want to do some more work on it? So that we continue to grow our net, expand our reach, so that we can share this information with the people developing the drugs so that we can, fast track things through and get into a trial. And I also am really big on communication. I want to establish this communication standard with the community. I am fairly responsive. I'm fairly responsive to email, but I'm also part-time. I'm 20 hours a week. So if you don't hear from me right away, that doesn't mean you'll never hear from me. That means that it's on my to-do list. I always have a big to-do list, and I will get back to you, because one thing that I can't stand is when I send an email and I don't get a response. So it is coming, and I want to make sure that when I send out these newsletters at the end of each month, it's expected. It's not a surprise. I know we all get inundated with emails. I know that social media is on our phones and we all scroll, but if there's something that is expected and consistent, it can be planned for and hopefully even looked forward to. So now at this stage, we're here, we're educating, we've done the soft launch, but by the end of the hour, I hope that we are here and we are actually doing these things. So my big kind of tagline that I've been saying since I joined the Alliance is that hope and reality can coexist, and they already do, and we want to keep that the norm. So the last half hour, I did pretty good staying to a half hour for my little presentation. The last half hour we have for question and answer. As noted by Carrie in the beginning and in the letter, we asked for all questions to be sent ahead so that we could keep ourselves on pace. But if you have a question or if something new came up as I was talking, you can send it to research@Dup15q or to info@Dup15q, and we'll compile them. And then I plan to release an additional FAQ document at the end. And Carrie unmuted, so she wants to say something here.
Oh, you're a good watcher. No, I was just going to add in. Did you finish your comments? And I'll just add in a couple of questions.
Questions for me?
Oh no, just for the group. Did you
Oh.
finish what you were going to say?
Yeah.
Okay, great. So I just wanted to, and feel free if anyone wants to add in more questions. You can also private message myself if you're not wanting to put it out to the whole community. One of the big things I think people forget, and I want you to touch on it a second, because I don't know if we went back to the longboard data and how important that was. But this is, I know sometimes you guys get inundated with surveys, you get inundated with questions. We say, sign up for Ladder, for Citizen, for this and this. And I actually was just on a call this morning with another community about this. If you're ever unclear and you're like, where is this going? Like, what is happening with this? I'll just give you a real world example that Ryan and I were in a meeting one day, and if you all are in Ladder, which is our patient registry database, in case you're like, what is Ladder? We actually had a pharma partner say, talk about LGS. And I said, do you want LGS data? I can tell you how many of our patients have LGS. And I said, we just sent out a 1-question survey and 137 of you answered. And he was like, absolutely. We want to know if you guys could be on a trial for LGS in the future. It's that one question that you may go, why are they asking this? That we can't always share the backstory, or we can't always have the capacity to share all the details. But when you see surveys, know that they're coming from a place of either interest, they're coming from a place of someone saying, we could have interest. Jackie and I have had conversations with pharma partners before Ryan came on for 2 years. Sometimes they come up, something comes of them, sometimes nothing comes of them. So we don't want to get the community's hopes up either because we'll get excited and we'll say, oh, someone's interested. James, I'm curious, not to put you on the spot, but can you express how important the community's buy-in and involvement in data is for a company like where you work? Because you've seen this progress, you see how long things take, but I want people to understand that that 1-question survey can make a big difference. Do you have any other feedback on that may encourage people why those are so important?
Sure. I think this probably falls into the learn bucket, which is the foundation for everything. We can't do anything actionable if we don't know anything. I think there's a saying about sort of wars being lost not because of lack of soldiers, but lack of information. And I think this is true for sort of fighting disease and treating disease, is that a lot of the disease treatments, for instance, Alzheimer's, one of the reasons those types of disease are very difficult to treat is we don't always know everything about the disease. The more we can know, the better we are in terms of developing therapies. So I think answering questions, inputting data, participating in studies that clinicians and scientists can learn all the little subtleties of the disease. Another thing that I can mention for people who are interested in, we were talking to SCN8A. It's another developmental epileptic encephalopathy. So these patients have severe epilepsy and other symptoms, very similar to Dup15q. It turns out there are actually lots of subgroups of patients depending on their symptoms. So they all have the same gene, but even with the same gene, they are broken down based on their symptoms into several subgroups. We wouldn't know that if we didn't have data, and that informs clinical trials. You might decide that this drug would be best for groups A and B, but this other drug would be best for groups C and D. We can't know that until we have learned as much as we can. So to Carrie's point, highly support participating in whatever you can and however you can. I know it's always a challenge with Dup15q patients to get them to clinics, and there's a different reality than to say that than to do it. But when you can, I encourage participating. And then the other thing I would say, and holds true for today, is don't be scared to ask questions. Hopefully I'm not intimidating, and neither is Ryan or Jackie or Carrie. But I think the more questions you ask, if one of the reasons you aren't participating is because you're not sure, or you're scared, or you're nervous, you're not sure what's going to happen, just ask questions, and people are often very happy to answer. And if those people aren't happy to answer, bring them to the Alliance, and myself and Ryan will be happy to answer and help. And so I encourage asking questions today, and when participating in studies or seeing announcements about studies, I really highly encourage you to ask. And then that allows us to learn more about how we could better communicate. The more we get certain questions, we realize we haven't communicated the right way or effectively, and we can adjust for the future.
Yeah. So that kind of aligns with the next kind of phase here. So the questions and answers, you guys provided them early, and there were a lot of themes, which means that we needed to explain things in a different way or present them as part of a larger story. Because sometimes if certain terms are just thrown out there with no context, that makes it really difficult to digest and understand. So one of the things that you've probably heard about that I've mentioned a couple times now and promised more of an explanation is the natural history study. So I put together 5 FAQs. The professor in me is dying at the amount of words on these slides here, but this is a dual purpose presentation where it's going to live on the website. It can be used for referencing later. So there are a lot of words. I am sorry. I'm not going to read them all, or at least I'm going to try not to. But I'm going to lie and read this right now. But a natural history study is a clinical research project to assess health outcomes without changing the participants' routine. So essentially, it's an observational study where data is collected over a period of time. It is monitored by a team of clinicians using a standardized protocol. So a lot of the assessments that you all might be used to already when you go in for care, but they're documented over time to track progression. Because right now, we don't know what the natural progression of Dup15q is. We don't know the nuances between interstitial and isodicentric Dup15q. There are shared symptoms, seizures, GI, behavior, autism, but do they change? When do they change? Do they always change at the same time? Is there anything that can predict when they're going to change, how they're going to change, etc.? So in order to find answers to these, we do what's called a longitudinal, you can think longitudinal long-term study of a given population over time using the same measures. Now, some of the questions is, why do we have to do this? Why can't we just pull the data from clinicians? Well, the standardization is important. And also, how does it benefit the community? Well, you can identify new biomarkers. And if that's just another word you're not familiar with, stay tuned. It's the next slide. You can identify new biomarkers. It'll establish a baseline for drug efficacy. Natural history studies are very important when it comes to developing drugs and seeing whether or not they're working well. Many times from my role here and in my past life talking to people, they say, oh, well, there's no natural history study data, so we're kind of stuck right now. Or there's no natural history study data, so how are we going to be able to tell if something's working? It's kind of, this is not the greatest analogy, but it's kind of like if you've never had pizza before and someone gives you a lukewarm piece that's been sitting out for several days, and then they sprinkle cheese on top and say, does it taste better? You have no idea. You don't know what good pizza tastes like. So in order to understand if something's working, you have to know what's happening in a standardized, non-interventional space. It will also allow sponsors, these drug developers, these biopharma companies, to understand what we need, to understand what will actually make a difference with regard to disease progression, as well as quality of life and disease burden. It will reveal patterns, progression, key characteristics that meet new criteria for clinical trial enrollment. So as Carrie was mentioning, that LGS survey question and just having this collection of data might show something that might make our community eligible for another potential clinical trial later down the line. It will help us, again, identify those genetic subtypes, create better endpoints, and inform the design of clinical trials for when they come up. You don't want to just measure everything all the time. That's overwhelming, that's tedious, and that sometimes you can have too much information. So it has to be aligned and tailored effectively. And it can accelerate recruitment for downstream interventional studies, meaning that if there is trust within the community from getting these data out there and you actually see movement and we report back to you, which that bidirectional communication is essential, then we can continue the ball moving forward down the line. So one of the pieces that came out was on natural history study on the social media, but once I'm done writing this, it is being submitted to the FDA in February. We currently have 3 PIs who are interested and who are going to run the trial, with additional PIs at various sites across the country who are going to be enrolling for this. It's moving forward. We're doing it. Whether or not we get this FDA money, we are going to lean on our industry partners and donors to get this off the ground, because we know how important it is. Okay. So one of the things that came up is biomarker, another word that's just thrown around. Well, biomarker, something that can be measured. Some examples of biomarkers: blood pressure, cholesterol. If it changes, that is usually an indication that something is working well or something is not working well at all, or that you can expect a major change down the road. Cool. So if we find more, how will this help our community? Well, it can be used to indicate progression or status. Drug developers can use them to test and see how effective their drug is at a given point. It will make designing clinical trials more straightforward. You watch these few things over time, you know if it's working. And then when you go, when these biotech companies who are developing the drugs go to ask for more money to launch big time trials, they can show them these data, and the investors, who are the people with the money, they will say, I love this. Here's $1 bajillion. And that's how it always happens. No, I'm just kidding. It's way more complicated than that. But an example in our community that's been really effective and that has really helped get the clinical trial situation happening is the EEG biomarker used in the Roche trial. That would not have happened without this community, because the clinicians who work with our Dup15q family members, they noticed that there was something interesting on the EEG. So they launched a small pilot study, which some of you may have been enrolled in. So thank you. Once that data was taken in, analyzed, and published, then it became a standard. Then it became a real live biomarker that everyone knew about, that could then be used to track whether or not something was working. Roche, they came across the biomarker. They had a drug that wasn't working so well in something else, but had the potential to make a big impact in our specific community, and are using this EEG biomarker to launch the first Dup15q specific clinical trial. This is gigantic. This is huge. Everyone should be very proud. Additionally, we're not done.
One more thing, I hadn't remembered this earlier, but this is actually another example of a seed grant. Now, there was lots of different money that helped fund this. This is a big project that the Just group participated in, but I do know there was another funded research project, a seed grant that the Dup15q Alliance provided in support of this work to one of Shefali's students.
Yes. And also, just to kind of close out the biomarker, there are also molecular biomarkers, which could be molecules in the blood or in cells or changes in gene expression that can be used and trackable over time. So those of you who gave blood for the Combined Brain Initiative this summer, you're already making a very large contribution to this effort here. So keep up the good work. Thank you so much. This is really a group effort. Thank you. So speaking of drugs, what's the difference between a novel and a repurposed drug? Novel, sometimes you might see de novo. They're new. You could think novel, new. And I will leave these fancy definitions here for review later in your life. Repurposed is something that has already been approved by the FDA, and based on what they've seen with it, it might be a good candidate for something else. Okay, so kind of like another terrible analogy, I have this coffee mug. It is meant to hold coffee, but it could also serve as a watering can, or it could also serve as something that holds water. You know, it's made for one thing, but it could be used for something else. So to kind of break down the difference for how this might look with regard to our efforts here to get a drug into people, novel drug discovery and the timeline from discovering, screening, optimizing preclinical studies in mice, cells, rats, non-human primates, then into humans, clinical trials, then safety, etc., FDA review and licensing, 10 to 17 years on average. Could be a little bit more, could be a little bit less, could be a lot more, could be a lot less. Everything is approximate in this space. But if you have a repurposed drug, something's already been through, you see my arrow, something's already been through all of these phases. And now it's been approved. The FDA is like, yeah, this should be fine. But then it has to be acquired by the interested parties who want to then test it in the specific disease space. It will be revised, optimized, changed for preclinical development, and then it'll get into people. So this shortens the time frame from discovery to people. I want to say significantly, but it's pseudo significantly, I'll say. So that was a common question. What's the difference between novel and repurposed drug? And I know Jackie was going to speak a little bit about what this means to the parents in the community. So go ahead, Jackie.
Yeah, so I think for a key thing about repurposed drugs is that it shows there's some safety data behind it. And so for a lot of our families, that's a really comforting thing to have in your back pocket, to know that a drug that is FDA approved has gone through rigorous safety data. And for our families, again, I think that that's really kind of a source of comfort, I guess, as we're entering into clinical trials. I don't want that, however, to take away from some of these novel drugs. I think that that's where we're stepping into the realm of the cure of these disease-modifying therapeutics. They are novel, but they are showing, you know, some significant progress in other diseases, not in Dup15q. So, and they also go through rigorous safety data or safety testing as well prior to going into potentially into our children. So I think it's for me, it's very exciting for both avenues to be happening, but I can certainly understand and appreciate why for some of our families that repurposed drugs just have that extra layer of comfort and, you know, safety behind them.
Yeah, and just to kind of add, some of these novel drugs, they're already here in preclinical. So it's not that we're starting back at year 1. Many of them are already maybe in year like 7 to 10 plus. But those data are always evolving. I only have 2 more because I know I have a hard stop at 1, and many of you do as well, probably eating lunch. So I'm just going to keep on rolling through. How will clinical trial participation help the community? Well, here are my definitions. What are the benefits? Potential for identifying curative interventions. Number 1, help to expand the treatment indication for Dup15q. So if you have something considered an indication, that means you have access to something. And Jackie, I'll give you a second to talk a little bit more about that, but I just want to run through these few to get to the clinical trials that we are in right now. We can't learn anything if we don't do the analysis. So by enrolling in these clinical trials, you're giving us the opportunity to learn about your child and about the disorder at large. So when we have that information, we're able to engage, attract, and retain interest from these drug developers. And then it will expand the treatment profile for the future generations of those diagnosed with Dup15q. The Quindesem or Roche trial right now is still enrolling. I know Jackie has been personally calling people to encourage them to enroll, so pick up her calls. And then the Pacific study, which is a longboard study, it was a unique one. In that it was a basket trial, so it wasn't Dup15q specific, but many Dup15q individuals meet the criteria. Data just came out on Monday, and it was super impressive. It showed, I believe it was 61% of what's considered other DEEs, which is developmental epileptic encephalopathies, I think. It showed substantial improvement when compared to the placebo group, which is the non-treatment receiving group. Really, really exciting. They have, for example of how exciting this is, they ended the day yesterday with over $200 million of investments so that they could push forward to the next phase of clinical trials. I think it was $242 million, if I'm being exact. But this means that they can keep refining this and then eventually potentially optimize it for our community, or that they're learning that one specific intervention could have a broad impact on a community or communities that share specific symptoms. So it's huge. Carrie, did you want to say something about that?
Yes, and we actually have people sending in questions right now about it. One of the big things in that, and I'm getting this in the chat as well, is it is so important that we're participating in these trials. One of the things that the companies make very clear to us is if a community is excited, if a community, the voice of our community drives whether a company is interested. And we'll get at, Jackie and I, before Ryan has gotten here, have been asked, tell us how the clinical trial participation is going. Like even other companies will ask. And so don't forget, if you are interested in the Roche trial, to make sure that, I know there's still some spots at Rush that are available. There's still some other sites opening, but it's really important we show up. And I know some people are nervous. Remember, when you call, you're not registering right then. They're great. They'll answer questions. They're going to talk you through it. They're going to talk to you about talking to your current providers. But it is important we show up for these, or the interest changes. And so we want to make sure our voice is really loud in this. But I just wanted to put that plug in. If you've been hesitant, it's okay to call. We'll never convince you one way or the other, because your child's safety and your decisions are your decisions. But we want to make sure we're really showing up for this.
And there are some other questions in the chat, and we're going to collect those because we are coming up against the end of time, but you're not just throwing them into the void. We will get back to them. We will respond to them, hopefully by the end of next week at the latest. So the last question is the mouse. As I mentioned earlier, you all have heard about mice. I know that there have been some seed grants for mice that have then went to feed forward into really gigantic grants from the NIH. So why do I care about mice? Well, a lot of reasons, because it's a great way to understand what's actually happening at the molecular level after a duplication occurs in that specific region, which genes are responsible for which features. And once we understand those things, we can then use them as targets for drug development. We can also understand how duplication may affect certain behaviors, and again, link them back to targets for potential drug candidates. Preclinical assessment of drug efficacy. It shows that the drug works. And that is really important, because that is what will convince investors to fund the development, the build out of a specific drug program that can eventually lead to a clinical trial. I have close connections to people in the investment space, and I said, okay, you're an investor. What does a mouse mean to you? And they put down what they were doing, and they said, everything. It's like, if I only see stuff in cells, that's really exciting, but I want to see how it works in a living model organism. I want to see if there is a difference in baseline after the drug is there. And if they do see that, then they're more likely to invest, because if the drug works in a mouse, it's more likely to work in humans. Nothing is guaranteed in science. You will very rarely hear me say the word prove, but it strongly suggests that something will work in a human. So it is that foundation, it is that extra support, it is that validation in a mouse. So there are a few different mouse models for Dup15q right now that are being developed, and it takes time. Building a mouse can be an entire PhD project, and an average PhD project is 5 to 6 years. And then once you have the mouse developed, you have to do all of the other assessments. So we're very fortunate right now that we have a handful of mouse models that are undergoing these preliminary preclinical assessments that can then be used to test some of these other preclinical drugs that are in various stages of development. So we have a lot of the puzzle pieces in place, and I believe that we're at the space where we're now putting it together to make a really cool picture. So we started off by saying that we're going to expand the Alliance's scientific footprint to change the reality of a Dup15q diagnosis. And I did an upside down triangle, if you were wondering why it was upside down, so that it could point to the fact that if we work on these 3 areas, we can actually do that in the future. Step by step, any progress counts as progress. Setbacks are normal. They're not periods, they're commas or a semicolon. And it just gives anything you learn is something that you learned. And that's all I have for today. Thank you.
Ryan, thank you. I think that folks watching will be impressed that you've caught on so quickly to so much information. We're so grateful that you are brought to us. We're so glad to have you on the team. And Jackie and James give endless amount of hours to help us and to help cover those other hours where we can't quite get you just yet. So thank you for a fantastic presentation. We do see the questions come in. I do want to address one because I've had it several times. The Roche trial cohort 1 does still have openings. People have said how many are in there. We actually can't share the actual exact numbers, but we can tell you that there are a few spots left, and then we get to cohort 2. So hopefully, we need people to enroll in that. And remember, that's for kiddos with seizures. On our website, and this is not it's not a seizure indication though. Don't forget that. We've talked about that on a couple of our webinars. Always, you can see on our website under our clinical trial section, the updated folks to get in contact with the different locations, the ages. A couple of the locations have applied to get the age increased. It's always up to date as soon as we get those. Jackie, Ryan, and James are probably my first note. They're our first folks I send the information to. Then we send it out via text. Also, if you haven't signed up for our text alerts, NACA sends those out also. We really try to keep that to research, to keep you hot off the press news. So we're not just blowing you up with like, buy a t-shirt. It's not that type of information. So NACA, if you'll throw in the chat how people can sign up for our research text alerts, that's, hey, this trial site just opened, jump on this. It's pertinent breaking news for us. So make sure you get on those as well. We have 2 more minutes. Jackie, James, anything else we missed?
No, but I will take another like 15 seconds just to reemphasize how important this community is in this effort. Science and research, which drive treatments and disease modifying therapies, require money, both for the seed grants, for the biomarker, for all of the things that Ryan has talked about. And so, we only have so much reach when it's just 5, 6, 7 people. So we need the entire community to really get behind and support this and help us raise the money that we need, because this is starting. We have the gala coming up, and we're really excited for the progress we're going to make. So thank you all.
Absolutely. Jackie, anything from you?
No, I'm just so excited. I'm excited, and I think Ryan's tagline is the most is really important, the hope and reality, because I'm always a cautiously optimistic person, but I've been doing this for about 2 years since my daughter got diagnosed, and I've never been more hopeful. And I think, but there's the reality behind it to support the hope. So there's really amazing stuff happening, and like James is saying, we just kind of need to find the right people and find the funding, and I think we'll have some really exciting outcomes for all of our individuals, whether they're babies through adults. So we're doing the best we can to help everyone.
Yeah, thank you, Jackie, and thank you for your leadership on the board and as a co-chair with James on the science committee. And then thank you to our technical support, who is always in the background taking questions. NACA is fantastic. Appreciate her being behind the scenes and all that she does for the community too. So thank you all. If you have questions, again, if you don't know how to reach us, again, info@dup15q.org. Is always an easy way, or you can do the same thing and put research@dup15q.org. And there's a team who actually answers the research emails. You're not getting medical advice from that. I know we talked about that a little bit, that Ryan's a PhD. We've gotten a few MD questions, and we try to be really careful about offering medical advice, but we can always feel free to send those. We can point you in the right direction, hopefully, if we know anything or know of a doctor who specializes in something. But just wanted to clarify that and reiterate that again from her talk at the beginning. So thank you all. What a great way to start the new year. Hopefully we'll see all of you at the gala. Wouldn't that be great, Jackie? We would love that.
Yeah, so exciting. It's going to be a great time. It's a beautiful venue. So check out online, and there's tons of information, and you can buy tickets and join this wonderful crew here and everybody else who's coming. So
yeah, thank you. Thank you all so much.
Thanks,
and happy
everyone.
New Year.
Bye.
Thank
Bye-bye.
you. Bye.